The viral DNA or bacteriophage, whichever is infecting that cell, it inserts its DNA or RNA into that cell, and the Cas proteins recognize that that DNA or RNA is different from what's normally in the cell, so they grab it and then package it into these small spacers, which are then inserted into the CRISPR array, which then, once they're inserted, they are then transcribed into CRISPR RNA or cRNA, which is then attached to a different Cas protein. So the CRISPR RNA acts as a guide for the Cas protein to then target the specific invading genomic sequence, so that way it can then be degraded and expelled from the cell.
The epigenome is the series of chemical compounds that influence gene expression without altering the actual DNA sequence itself. And what alters the gene expression is environmental factors, which silence or express the genes using modifications such as methylation, which is the addition of a methyl group, or unmethylation, which is losing methyl groups. There are also acetylation and histone modifications, but the most common one seen is methylation.
Different people have different genes, and certain drugs are more effective on them depending on the genes they have. So if you have like a certain mutation in your genes, then a drug might be harder to metabolize, and it might make a certain drug easier to metabolize. And this is why certain treatments aren't that effective on patients, because it's not like the genes won't like really let the patient metabolize the drug as well as other people would.
And it is known that without the expression of ACKR1 on the erythrocyte precursors, hematopoietic stem cells differentiate into neutrophilic granulocytes with special properties. One of these properties is that these neutrophils readily migrate to the tissues and spleen, which accounts for the neutropenia because they're not found in the bloodstream anymore. They're found in the spleen and tissues. But another one of these properties is that scientists believe that these neutrophils provide a positive impact on the innate immune responses. So these neutrophils actually create a stronger immune system, which can be good and bad, because if you have something you need to fight off, you're probably better off at fighting it. But in an overactive immune system, you could have like acute inflammatory disorder or like allergies that you might develop.
So scientists believe that the shocking development of developmental genes increasing in production after the death of an organism is due to the body condition looking very similar to that of an embryo. So soon after an organism dies, the body condition looks similar to an embryo, so the body starts acting as if it is an embryo, so it increases production of genes and cells. The gene information can also be used to predict quality of organ transplants, because if you know how genes react after the death, then you can see how a new organ will react when it's implanted into a new body. And it can also provide accurate estimates of the time of death based off how many genes or what genes are ramping up after death.
One of the process of HCSC is that the immune system gets completely destroyed. And when that happens, the person who's diagnosed with this therapy, they are used by chemotherapy reagent, and what happens after that is usually the patient usually goes from a state of having their immune system completely destroyed to having it be rebooted or reprogrammed in a way for it to function normally. That will kind of prevent further MS activity.
Since this is such a new study, there's not much out there, but what is out there shows that epilepsy is almost definitely not linked to a single gene, be it that in one person versus another person, or within one person there's often linkage to multiple genes, which makes the whole picture much more complicated. Now, current treatments are not quite there yet. We're currently dealing with neurotransmitters such as the GABA neurotransmitter, and scientists are looking at the relationship between these neurotransmitters and brain cells and seeing how they interact and how they can cause these neuronal misfirings.
In order to understand when this microbiome is out of balance, we need to understand what a healthy microbiome is, and that's the purpose of the Human Microbiome Project, to establish what a healthy microbiome is and what our core microorganisms should be. When this balance is offset, it can correlate with disease, as in the case of colorectal cancer. Now, scientists have a pretty firm understanding on what colorectal cancer is and how it progresses and the genetics of it, but we do not yet know why these mutations and why these tumors arise in the first place. However, it's been seen in some research that this one particular bacterium, Fusobacterium, found in most human intestinal tracts, when it's overly abundant, it can induce tumors to grow because it feeds off of tumors. And interestingly, this bacterium is 12 times more abundant in those with colorectal cancer than those without.